Hello, Dan Guerra here. This is another VerEvMed recording. Today we're going to take a little bit of a exit from what we have been doing. We're going to talk about cancer immunotherapy and how that, interestingly, knocked on to vitiligo treatment. Now, if you don't know what vitiligo is, well, it's okay. We're going to talk about it. But I think all of us have heard about cancer immunotherapy. It's one of the three types of therapy used for cancer. One is, of course, surgery. The other is, well, actually, it's one of four now. Sorry. First is surgery. We'll say surgery is first, even though it's not usually the first line of defense. Second is strict chemotherapy, which has to do with very harsh chemicals which destroy tumors. The third is radiation, which, of course, irradiates the tumor and ablates it that way. And the fourth now is immunotherapy. Immunotherapy then is the fourth armamentarium against cancer. It's becoming much more prominent in many cancers like non-small cell lung carcinomas. It's also being used more and more in bladder cancer, prostate cancer, breast cancer. So all the major players. Immunotherapy basically involves using the immune system to attack the tumor cells. So we're going to get started right now, and we're going to talk a little bit briefly about just this whole immune cell repertoire, very briefly. This is not going to be any kind of primer or strong education for immunology, just to kind of get us nested into the topic. And then I want to talk about a couple of very important so-called checkpoint inhibitors in cancer immunotherapy, and then how that relates to vitiligo. Okay, so let's get started. Again, I'm Dan Guerra, and I'm from VerEvMed. And what we do is we take deep dives into the literature, scientific literature, and we provide for our clients an explanation, a synopsis, and a derivation into areas of research derived from that, and verification of the evidence derived from published literature. So that's what we do, and we very much appreciate anyone that wants to contact us so that we might be able to work with you. So let's get started. All right, so let's take a look at these cells. So this comes from this particular link here, this diagram that does. So if you think about a tumor is going to have various kinds of antigens, okay? Antigens that are being generated by that tumor mass. Those tumor antigens are actually going to play a major role in whether or not the tumor will evade the immune system or will indeed activate it, and the tumor then thus get ablated by natural immune responses. Now, we can help that immune response along by using immunotherapy, and that's what we're talking about here. So the tumor produces these antigens. The antigens are picked up by an antigen-presenting cell, for example, a dendritic cell. So tumor antigens are picked up by the APC. They then present that antigen either with MHC class 1 or MHC class 2 surface proteins. The MHC class 1, that's the major histocompatibility complex, right? Or HLA in humans. It interacts with a T cell called CD8, so that's its cell surface marker, CD8. Naive, meaning it hasn't yet to be activated or differentiated. Whereas the MHC class 2 will interact with a naive CD4 T cell. Now, these are two different T cell lineages that come directly from their precursors, of course. If we engage CD8, we get what's called a cellular immune response. We make cytotoxic T lymphocytes. Some of those will go to memory, and some of those will directly attack the tumor. Okay. Also, what can occur is when MHC class 2 interacts with naive CD4 T cells, we go through the Th1, Th2 cell lineage. The Th1 cell lineage can also produce cytotoxic T lymphocytes, but it also produces a lot of cytokines and chemokines. These are all going to all be pro-inflammatory, and they're also going to engage other cell lineages to amplify an immune response against the tumor, specifically tumor antigens that were derived via the antigen-presenting cell after digestion and then complexing with MHC and presentation on its surface, right? Th2 is also involved, another cell lineage of the CD4 lineage. They'll produce another suite of cytokines, 4, 5, and 13. Those are interleukins 4, 5, and 13. But also, very significantly, Th2 reacts with the B cell lineage. It's going to activate B cells and turn them into plasma cells. Those plasma cells are going to produce IgG. In other words, it's going to make antibodies, those glycoproteins known as antibodies. And those antibodies are going to be specific for, way back here, those cell surface markers, those tumor antigens. Okay? And that's known as the humoral immune response. This is all then the acquired immune response. And this goes back to the innate immune response, okay? And the innate immune response is a lot of other things that we're not showing you here. So that's your basic players. So what are we talking about here in terms of specificity of immunotherapy? Well, there's a protein called programmed death 1 or PD-1. It's a so-called type 1 membrane protein. It's about 268 amino acids. Now, PD-1 belongs to a superfamily of other proteins that are called CD28 or CTLA4. And it's a huge family of surface cell proteins that act as receptors for the T cells. And not only T cells, but other immune cells and indeed other cells entirely, such as endothelial. Now, PD-1 structure includes an extracellular so-called Ig5 domain. It has the transmembrane, the spanning transmembrane region. It also has an intracellular terminus, which interacts with everything in the cytoplasm. So it's your typical membrane-bound receptor. So PD stands for programmed death. So you get the idea that programmed death means that when this thing is engaged, when PD-1 is engaged, it's going to kill cells, okay? But it'll kill itself if it's engaged the receptor on that specific T lymphocyte, for example. So PD-1 is paradigmatically found on the surface of activated T cells, B cells, and macrophages. However, it's also found on other cell lineages, and that's very significant here. But later you'll see. Now, both CTLA4 and PD-1 negatively regulate the immune response. That means they suppress it. They suppress it at the level of the T cells that have those surface proteins, those surface receptors. Okay? So let's go into a little bit more detail here. It's a paper that just came out in the Journal of Biomedical Science just recently, about a few weeks ago, actually. I'm taping this in July of 2017. So you've got an antigen-presenting cell or even a target cell. Doesn't matter which one, but for discussion here, we'll act as if it's an APC. And that could be a dendritic cell, for example. Dendritic cells like to talk to T cells. So they pick up a bunch of antigens. They're bound here to the MHC complex. That MHC is going to interact with this T cell receptor. Here's your T cell, naive T cell. But the T cell is also going to have a lot of secondary proteins or activator or suppressor proteins, other proteins that need to be engaged for the first signal, that is MHC bounding antigen to the T cell receptor, for this T cell to be activated. So you have two signals, basically. You have this classical canonical signal, and then you have a second signal. The second signal is far more elaborated, and it's going to involve suppressive motifs and stimulatory motifs. So if it's a suppressive motif, it's going to go in kind of a gradated form. These are the names of the proteins that are found that are going to engage the proteins on the antigen-presenting cell with the T cell. So you're going to have all of these various proteins, LAG3, TIM3. Yep, there it is, PD-1. Okay. Now PD-1 is going to be on the surface of the T cell for this particular diagram, and the PD-L1 and PD-L2, those are the PD ligands. So those ligands are going to be the proteins that ligate to the receptor. So that's what they're called. So we actually have in immunotherapy for cancers. For example, bladder cancer or non-small cell lung carcinoma, we have actually antibodies to the PDL and the PD. That means we knock down this suppression at the T cell and we knock down this suppression at the ligand back at the antigen-presenting cell, or even indeed the tumor cell, because the tumor can also present this. All right. Now, last of the suppressive lineages are CTLA4 itself, where the family gets its name. And it's going to interact with the cell surface marker on the antigen-presenting cell called BC1 or BC2. Okay, so that's when you start moving away from suppressive to stimulatory. So actually, this becomes stimulatory. These are all stimulatory signals from the antigen-presenting cell, and these are all the receptors for it. So CD40L is a very well established one for CD40 here, okay? So these are, again, cell surface markers are going to stimulate the T cell, and that's what you want when you want the T cell to kill the tumor cell, right? And this is all interacting, again, with these cell surface proteins, these tumor antigens, for example, that got secreted and loaded onto the MHC on the antigen-presenting cell, derive a signal through the T cell receptor or through the secondary signal, and either suppress that signal, so these T cells are resting, they're standing down, or they're turned up, and then when this turns up, that's the fully engaged T cell. So T cells then, we say, have immune checkpoints, and those are PD-1, for example, shown here, and CTLA-4. They reduce the autoimmune response against self tissues. So what they're there for is not to let tumors grow happily and kill you. They're there to suppress or prevent the immune response from destroying self, right? So that's what they're there for. That's why you need a secondary signal. It's called a co-stimulatory, co-suppressive signal. So it's like, yeah, we're ready to go ahead and respond to this and kill whatever brought this on board, whatever cell lineage brought this on board, but we want a second look, right? We want a little bit more evidence, and that more evidence comes from that secondary signal, right? And the secondary signal is nuanced, as I just said. All right, so we call those checkpoints. I don't particularly like that term because I think checkpoints, I think of cell cycle, and that's not really what we're talking about here. We're talking checkpoints on T cell activation. It's a subtle thing that when you get into this literature, you got to keep in mind. But it means the same thing. Checking point, meaning yeah, go or no go, basically like a checkpoint. So while most cancer immunotherapies accelerate T cell activity, immune checkpoint inhibitors, again, that's what they're called, release or repress the immune system's inhibition to instantiate anti-tumor immune response, okay? So they allow for these things to be removed, okay, or to be prevented from suppressing. And because they prevent from suppressing, you get activation of the T cell. That's what those kinds of immunotherapies are when we call them immune checkpoint inhibitors. If that's not clear, you'll see it in a minute again. And I'm going to describe it. So PD-1 is mainly expressed on activated CD4 T lymphocytes and CD8 T and B cells in the periphery. So this isn't talking about the center of origin of these T cells. It's talking once they've been mobilized and sent out to the periphery. It's an important thing in terms of the clinical applications of this immunotherapy, which we won't go into this morning. Since PD-1's expression is indeed induced, so this is an argument for what I just said, it likely regulates late phase immune responses, that is effector phase, memory response, chronic infection, these sorts of things. And that's happening in the periphery and not because it's an induced system, the early induction phase of the lymphoid organs, okay? Because it's something that looks like it's subsequent to the production of those naive cells, right? So PD-1 deficiency, actually, when you don't have a lot of that in just a healthy person, or actually a person that just has this deficiency, we're not talking tumors here, that's linked to autoimmune disease. Now, why is that? Because you have then a fully engaged T cell, B cell repertoire that's turned on, possibly with self-antigens that are not toxic to the system, right? So you get autoimmune cells. So a lot of times we're looking for in fact, the anti-PD-1 and PD-L1 are used for autoimmune diseases, right? We want to knock that down to get rid of autoimmune diseases. So, I mean, we want to, actually not knock down, we want to activate that programmed death, right? Because we don't want those T cells acting as autoimmune inflammatory responders, right? So you sometimes want to engage PD-1, right? And so there are drugs that do that, or in this case with immunotherapy, targeting T cells to turn them back on, remove that suppression, right? Here you want to use an anti-PD-1 or anti-PD-L1, because there you're going to get rid of that programmed cell death of the T cells, and you're going to activate your immune response. You see, it's a Janus system, like so many things in biology. So immunotherapeutics target PD-1 and thus enhance T cell ablation of the cancer cells. So you get rid of the programmed death in the T cell. T cell is active, it's dominant, it's potent, it's vibrant. It's going to kill cancer cells, right, with those specific surface antigens, those tumor antigens. So tumors can produce PD-1 ligand. They're pretty tricky. not just the antigen-presenting cells. The tumors themselves may be selected for or may engage in the process of making the ligand. So right away, the tumor, if it's interacting with the T cell directly, can shut down the T cell immune response. So really very nasty tumors, right? Because they're going to start slowing that down. It's shown here. It's a paper that comes from Nature Immunology, 2013. So here's an infected cell here. This is like a tumor cell, or for example, or maybe a virally infected cell. We'll say that it's expressing the PD ligand on its surface. Of course, all cells have the MHC complex. There's an antigen, it's interacting with the T cell receptor. Okay, we all know what that's going to happen here. You're trying to turn on this T cell. It's an effector T cell. We're going to say it's a CD8. Now, this tumor itself can produce chemicals that can shut down one of the factors associated with causing the T cell to be activated. This is called SHP2. So this is one of the targets in immunotherapy, right? Preventing this shutdown of SHP2. So that's one thing that's just shown in this slide here, not what we're going to talk about today. So that attenuates the T cell receptor if you attenuate that intracellular transcription factor. Now, normally what would happen is you'd get a lot of cytokines, and those cytokines would cause an inflammation, trigger a response of more T cells would come to the site and would kill that tumor, right? But it's all getting shut down by this ligand. So the important point on this slide here, on this part A, is PD-1 controls the effector phase of immunity, inducing unresponsiveness through attenuating the antigen-specific signal. So that's the antigen-specific signal down here. You're attenuating it by making that ligand bind to its receptor, the programmed death receptor, shutting down, standing down those T cells. Now, if you're thinking about the T cell itself, right, let's have it now expressing the CTLA4. Okay, it's another one of these immunosuppressive targets, right? So FOXP3 induces this production of this protein. Now, what happens when that occurs is that if you have a cell, like a dendritic cell, that has a depletion of CD80 and CD86, you don't engage this cell. Okay, you don't engage the cell. So CTLA4 controls the function of activated T cells. Okay, so that's what it's supposed to be doing. So CTLA4 dominantly captures normally CD80 and CD86. Okay, on antigen presenting cells. Okay, this is like a dendritic cell to me, and then it downregulates the co-stimulatory activity of CD28. Okay, this is an effector T cell. It's a T cell that's ready to go. So this is an inhibitory T cell, or like a Treg, T regulatory cell. It's doing this, and it's telling the dendritic cell, slow down the activation. So, okay, so it's going to react with these two proteins here. All right, so that's modulating then the effector T cell. This is the cell that's going to go and kill the tumor, or maybe because autoimmunity, right? And that's why the system is set up to shut down autoimmunity. That's why you have inhibitory or regulatory T cells to shut down effector T cells. Okay, so just a case in point, TGF-beta1 is an important cytokine produced by FOXP3 cells, right? There it is, TGF-beta. And it supports the growth and differentiation of these T cells and suppresses diverse immune responses. So again, there's yet another target that we see in the literature, TGF-beta targeting mechanism to try to either promote the regulation of T cells when you're trying to decrease autoimmune disease, or to shut down these inhibitory T cells when you want to promote T cell activation, killing a tumor. Okay, so this is a general consideration that we understand. This is only a few years old, and this particular literature has been refined a bit, but basically this is the basic tenets of it. All right, now it comes to our interesting knock-on. So this paper came out of JAMA, it's a Journal of American Medical Association of Dermatology. It was published very recently, just in July 12th. I can tell you that was less than a week ago from when I'm speaking now. All right, now PD-1, now we know all about what's going on here. PD-1 and PD-L1 are immune checkpoints, that's what they call them, that prevent the immune system from autoimmunity. Okay, clear on that. Blockade of those, of the PD-1 or PD-L1, prevents tumors from escaping the immune response. Hopefully we made that clear to you, right? So that's another important, just, by the way. Anti-PD-1, and here are a couple of monoclonal antibodies that are out there, Nivolumab and pembrolizumab. Those are the two drugs right now used in many immunotherapies in cancers. Nivolumab and pembrolizumab. Okay, now those are going to be targeting the PD complexes. Okay, there's one more that affects the receptor PD1 on the T cell, but there's also an anti-PD ligand one, and that is atezolizumab. So those three drugs are commonly used now for late-stage non-small cell lung carcinoma. That's one of the big players that's been using these drugs. And that's when you talk about immunotherapy. If you know somebody that's on immunotherapy, they're in late-stage disease. And what they're trying to do is activate that T cell lineage. Of course, the side effect when you do that is you might get an autoimmune response, right? You might get all kinds of other issues, like even dermatitis or any kind of itis disease, right? Any kind of inflammatory disease is going to be kicked up when you activate T cells, and activating them by shutting down the normal suppression. And that's what you're doing when you're using these monoclonal antibodies, right? Nivolumab, etc. All right. Now, interestingly, because I've been talking in the aging lectures about DNA repair, and we're going to get back to that. In fact, we're going to land right on mismatch repair. So, wow, how interesting that this is exactly fitting into this discussion. It's amazing. Anyways, mismatch repair mutations, which are common in tumor cells. Now, remember why that's happening. You got a lot of mutation in the tumor cell. It's generating a whole slew of tumor antigens. It's evading the immune system as any way that is possible by selection, right? By generating antigens, for example. And some of those antigens can actually be quite antigenic and therefore induce the immune response. And that's a good thing if you want to ablate the tumor. Anyways, this mismatch repair is common because you get a lack of good DNA repair in tumor cells. That's tumor cells, one of the calling cards, one of the addresses of tumor cells, right? When you look at a tumor cell that has a problem with DNA repair because it can't repair it, therefore it becomes mutagenized, therefore it becomes a tumor. See? All right. Now, what's interesting here is that mismatch repair mutants in tumor cell lines are particularly susceptible to PD-1 immunotherapy. Now, that tells you something interesting, right? So PD-1 immunotherapy doesn't always work. Now, that's too bad, but it doesn't always work. But what's really cool is we know that those tumors that are mismatch repair mutants, and we can do this by detecting satellite DNA, and satellite DNA is just a series of repeats. If we look at the series of repeats in satellite DNA coming from tumor cell lineages, right? Taking tumor cell and looking for satellite DNA that specifically has these repeats, looking to see if those repeats are being altered in their repetition. If they're altered in the repetition, it means you have a mismatch repair mutation. That is, mismatch repair, DNA mismatch repair is deficient, right? So we can then study the tumors from the level of their satellite DNA. We can say, aha, we got a lot of mismatch repair deficiency, MMR repair deficiency, and boy, look at these are our patients that are responding best to our anti-PD-1, our immunotherapy. Okay, so that's another thing to remember. Now, anti-PD-1 immunotherapy via pharmaceutical monoclonal antibody mechanism, the ones we just showed you, are shown to reverse loss of hair pigmentation in the disease called vitiligo. And vitiligo, if you don't know it, I'm not going to show you a picture. It's where you see a white patch of hair on an otherwise normally pigmented hairline. It can be black hair, red hair, blonde hair, whatever. Okay, so it's a complete loss of pigmentation. It's whitening of it, white hair, right? So that means there's no melanocytes, basically. And again, that's because vitiligo is associated with melanocytes in hair follicles. That's how you get your hair color in your hair. Now, melanocytes are indeed activated by pro-inflammatory cytokines and indeed by reactive oxygen. So that right there tells you something. If you have a suppressed immune response, right? Suppressed immune response, you may not get enough pro-inflammatory cytokines to activate melanocytes to cause pigmentation of the hair. I know that's kind of a mouthful and it's a long story, but I think you get the idea. Now that's one thing, and bracket that off and remember it. But there's more to it. Now that may account for localized hair repigmentation after you get an inflammation, such as folliculitis. Remember I told you about itises? So when you get an inflammation, it looks like, wow, people that had this vitiligo, all of a sudden that hair got repatriated, right? That is, it got repigmented. And that repigmentation is like, wow, what causes that? That's interesting. It looks like when you activate the immune response, right? So that's kind of interesting, because that's what an itis is, right? Folliculitis is an inflammation of the follicles, right? Hair follicles. All right. Now here's another interesting story. It only works in gray hair. So people with gray hair, maybe you can repigment gray hair. And this has something to do maybe with aging. So there's an inline there. And we're talking about DNA repair. Now we're back to even bringing up aging, which I admit I totally smuggled that into this talk. But I did it for a reason, because it all fits together. All right. Now, it works with gray hair because gray hair isn't non-pigmented, right? It doesn't mean there are no melanocytes. It just means there's very little melanin production. There's very little color pigmentation going on. Okay? Now, if you have no more melanocytes, like with white hair, like in actual aging hair population, which is white, then this doesn't work. You don't get the repigmentation. So again, that's melanocyte-specific vitiligo. So if anti-PD-1 actually cures vitiligo, and there are now therapies out there that are trying to use these anti-PD-1, which are normally cancer immunotherapeutics, monoclonal antibodies. Now they're looking to see if it can repair white hair and otherwise pigmented hair population. The most straightforward explanation of why this is happening, okay, from my point of view, is that cell death is averted. What cell death? Not the T cell stuff, the melanocytes. Because you know what? The melanocytes also have PD-1, okay? So it could be that the reason you're getting melanocyte ablation, removal, and therefore you're getting that white pigmentation in vitiligo, or you're getting the lightening in white hair as aging goes on, is because those melanocytes are going through programmed cell death. So maybe if you block programmed cell death by blocking the PD-1, PD-L1 system, hey, that's why you get repigmentation. So that way you don't even need to include lymphocytes as etiologic agents. So that's something I want you to keep in mind, just what I get when I read this kind of literature, okay? And it's logical. Okay? It's also applying a little bit of Occam's razor. Now, what is actually known about the pathophys of vitiligo? Here's a paper that I just found. It's not a journal I normally read. It's the Journal of Egyptian Women's Dermatologic Society, but it was a pretty good review, actually. It's a free online paper, so you can take a look at it by just jumping right into that link. All right, now, they bring up the hypotheses for the pathogenesis of vitiligo. So this is absolutely what you see in all the literature in vitiligo, not just in this particular journal, okay? So here they are, autoimmunity. So for some reason, the melanocytes are targeted, and so you get an autoimmune response by T lymphocytes, kills them, you get no pigment. Neurochemical poisoning, it seems like neural activity might be associated with vitiligo. So maybe it is the neurochemicals themselves, or lack of firing or alteration at the synaptic cleft that's happening in the nerves that are associated with hair follicles, associated with those melanocytes. And maybe that's inducing a kind of poisoning, okay? Third hypothesis is what they call, terrible name again, not mine, that's why I put in quotation or scare quotes, biochemical intoxication. They're talking about anything else that's not a neurochemical. So some other, like a peptide, or maybe a lipid, or some other kind of activating thing, like a oxygenated fatty acid, perhaps, like a prostaglandin or a leukotriene, maybe something like that is intoxicating the melanocytes, because you do get a lot of reactive oxygen in those cells. And maybe that's what's doing it, killing them. Fourth, sorry there's so many, is the antioxidant deficiency. That is, there's not enough of an antioxidant armamentarium, and so the cells die just by all of the machine gunning using reactive oxygen of those melanocytes, so they just get killed, right? That could be, and that's the fourth one. And then, of course, genetics or epigenetics, because some of this is inheritable, right? And also acquired via maybe some of those epigenetic linkages and lineages, which I admit are a lot of smoke and mirrors in some ways when you get at the 20,000 foot level. Or all of those potential mechanisms, which are here called hypotheses, could interact. Right? There's no reason why. No reason to rule any of those out. All right. So here's some final observations. DNA mismatch repair deficient tumors, okay? So that is, they don't have a good mismatch repair system. That means they make a lot of mutations. They're the most susceptible to anti-PD-1 immunotherapeutics. Now, they're not the only ones. And it depends on when you're making the observation on mismatch repair, when you look at those satellite DNA signatures, okay? So there's more to it. There's a lot of nuance to it, actually, in clinical oncology. But for now, I want you to just get that kind of take-home message. Now, that suggests, or perhaps, that several mutant antigens are formed with that MMR deficiency, because you're making lots of mutant proteins. You're making a lot of proteins that they're mutated because you have a lack of repair, okay? And that could cause a flood of T cell immune responses because you're making lots of mutant antigens. So therefore, you're arousing the suspicion of the T cell lineage. Now, because you have that PD-L-PD system, at the same time that's happening, the tumor cell is also launching and jacking up its ability to suppress the T cell. So that might be why you get that linkage with MMR deficiency and potency of anti-PD-1 CTLA4. Okay? So massive T cell response is maximized then by shutting down the suppression of the T cell activation caused by the tumor cells suppression of the T cells via the PD pathway. Okay, so those are my final observations of this system. So thanks very much for your interest and your attention. Have a great rest of July, and I will be back soon, getting back more into aging. There will be one more presentation. It's going to be a little bit of a different topic, but we'll get back to aging, and we're going to get right back into DNA repair as being associated directly with aging. So thanks a lot again for your attention, and have a pleasant